Today I'm going to talk about how did we end up using the Evosep system, what we're using it for, and how satisfied or dissatisfied we with the system. So just a few words about the PELAGO Bioscience. It's a CRO company located in Stockholm, Sweden. We mainly focused on doing CETSA, and I'll tell you more about it in a second. CETSA stands for Cellular Thermal Shift Assay. We're doing it in the targeted format with the help of antibody detection, but we also use it with a combination with a mass spec to elucidate the targets of small molecule drugs and more, of course. So just to stay on the same line, a few words about the cellular thermal shift assay. It's based on the very simple principles that the proteins, upon heating, undergo denaturation. So at some temperature, they start to unfold, and as they unfold, they become less soluble, aggregate, and can be removed from the solution. So for each protein, we can expect to see the characteristic melting curve. And if the protein binds a small molecule ligand, then it would change the thermal stability of the protein, which will be resulted in a shift of the protein melting curve. Thus, by looking at the protein melting curve with and without compound, we can detect the protein ligand binding events. What is so great about the CETSA is that it's not only working on the purified samples, the purified proteins, but it also works in the very complex systems, including the intact cells, tissues, and many, many more. We try it in bacteria, we've tried it in fungi, you name it, zebrafish. So when CETSA is combined with the quantitative MS-based proteomics, in one experiment, we can quantify and follow thousands of the proteins, which make it a perfect tool for drug target identification. The only thing we need to do, we need to incubate our cells with a compound or with a vehicle subjected to the heat challenge, remove aggregated proteins, quantify the soluble proteins, and then see which of the proteins got stabilized by the compounds, which got destabilized by the compound. And then we can easily visualize the results and find out the relevant proteins. And what is shown here on the top right of the slides is the results of the profiling of a number of compounds within one experiment, and a subset of proteins are shown along the x-axis, which show a destabilization or destabilization in any of these experiments. So in a drug target identification, we're really interested to look as deep into the proteome as possible to make sure we do not miss the target. That's why the good separation of the peptides is really key, but also each experiment takes quite some time, so it's the throughput is also important if we want to profile large number of compounds, as we can see here on the y-axis. Yeah, that's why it's a perfect match for the Evosep system to provide us both good chromatography separating peptides, but also robustness and throughput. So that's what we do, and I'm trying to change the slide and it doesn't.
So Alexey, I think if you use, now you have a laser pointer, if you use your arrows, I think you should move, yeah.
Okay, yeah, now it's moved. The CETSA is exciting, but once we heat our samples, removed aggregated proteins, what is left? It's quite standard quantitative MS-based proteomics. So we need to reduce our disulfides, we alkylate cysteines, digest proteins to the peptides. We use TMT labeling for the quantification. Then we do SP cleaning and two-dimensional chromatography. So first dimension, high pH fractionation. We end up with 60 useful fractions, and then we analyze them by online LC-MS. So in 2017, the whole MS facility at PELAGO consisted of one mass spec. It was a Q-Exactive HF, one nano-LC system, which was Ultimate, and the HPLC system from Agilent for offline fractionation. And so with this system, PELAGO started to explore the CETSA MA space, and then the number of projects grow pretty fast. So already 2018, we expanded with yet another LC-MS system. This time was HFX mass spectrometer and yet another Ultimate Nano LC. And it was pretty much the time when I joined the company, and we had a large increase of number of samples to run projects. So the uptime of the machines was really an important question for us. And so what we realized that we would really like to have yet another nano LC system because the main reason for downtime of these machines what the nano LC maintains. We either need to troubleshoot some leakage or change some parts or whatsoever. So 80% of failures were due to the nano LC. And we don't want our mass specs to stay idle while we're fixing the nano LC issues. That's why we thought, okay, we get yet another system and then we can hot swap the LC and keep running. So to ensure more than 90% of uptime and usage time of our mass specs. So we started to think which LC system we want to have. And then, so this is from the slides we had in 2018 with our requirements to the new nano LC system. And of course, it has to be comparable with our existing mass specs. It should be compatible with our methods and protocols we use in the company. It has to be stable and robust. And we wanted to be somehow different from what we had already, because the easiest solution would be just to get another Ultimate or AZLC, and it's easy to swap, easy to use, but most of the time this one extra system would just stay there and do nothing, and it didn't sound good. So we wanted to have something different with either new possibilities which we cannot get with our systems, or something completely different. And that time I heard quite some rumors about the Evosep, new way of delivering the gradient and so forth. So I thought it was a perfect moment for us to reach to the Evosep, to hear from them, and just to figure out if it's worth trying or not. Because what Evosep was advertised for during that time was throughput, application to clinical samples, and what we heard from Nikolai to reduce number of steps, and it was not very relevant to us. For us, what we needed is robust LC system for deep proteome profiling. And at that time, So we reached the Evosep team, and I'm trying to change the slide again. And it was easy to get in touch. Very nice communication back from Evosep team. We asked all our questions, whether it's compatible with our workflows or not, which allows us to go as deep into proteome as we used to do. And there were no ready answer for these questions, but what Evosep offered us is just a free trial. Get the system, try it for a couple of months, and then decide, is it what you need or not? And we decided, why not? And very quickly, the large box with Evosep system reached Stockholm. Was installed pretty fast, with almost like plug and play, filled with solvents, and ready to use. We set up pretty simple tests for us to understand if it's working or not. So we had some of our samples, TMT10 labeled. We processed them in our standard way with non-ultimate. When we first combined the 60 fractions into 10 in a checkboard manner, we used to use 2-hour gradients for individual fraction analysis, and it was almost 26 hours per sample on the mass spec. And we had an idea about the coverage we would normally get in this experiment. And then that time for the Evosep, instead of pulling fractions together, we just analyzed them individually, one fraction at a time, with a 20 minutes gradient, so ensuring 24 hours per sample. And we got very similar coverage, both in terms of proteins and the peptide groups, so it was a very good sign for us. So these are results here from the very first test. We did some more before we were convinced that this system can deliver. And we decided to keep it. So how we use the system today, it's almost 3 years past. I just took a screenshot from the system we have up and running in the lab right now. So we have very nice and lucky serial number 17. And total analysis we ran so far is almost 16,000. And I don't know, does it count all of them or only on the one mass spec? Because we move it around from time to time. And as you can see from the methods we use, our top favorite method is 30 samples per day. Majority of the samples we run with this method, and slightly less, we use 60 samples per day. So what we do now, we have our TMT labeled sample fractionated, and we analyze every other fraction, ending up with 30 fractions with 30 samples per day. We still stay at the 24 hours per TMT multiplexed samples, and we're quite happy with the coverage and robustness. Another things to add is that almost 3 years passed and we didn't have a PM yet on the machine. And I think it's not even close based on the number of analyses. And the system is just working. I think all over this time, we just changed the loop and one peristaltic pump, which is not peristaltic, but yeah, exchanged very fast and easy and very fast feedback from the Evosep team. So we are quite thankful. Also, probably important thing to add is that we love combining the Evosep LC system with the AZ columns from Thermo. So we use 15 centimeter column for both 60 samples per day method and 30 samples per day method. And in some cases, we also try the new extended method, 15 samples per day, and is also working fine, and we probably will try to use it more in our DIA label-free quantitation in the future. Yeah, so just a few examples of data which we can get if we have all the system up and running in a good way and in a good shape. So we had the internal R&D activities in the company, and one of the projects we decided to profile a large number of the compounds in the very same manner, in the very same system. We chosen HepG2 cells because our initial thoughts were to focus on the hepatotoxicity of the commercially available drugs and the compounds. And part of this study made this publication, which gave the title for today's talk, when we focused on 22 antiviral compounds out of these almost 200 compounds, and look at the binding profiles in the HepG2 cells. So we didn't focus on the viral proteins from the very beginning. So we were mostly interested in the host system response, and in particular HepG2 system. And by combining automation, so we have the Agilent Bravo robot and robust LC system from Evosep and fast mass spec HFX, we were able to design very fast and efficient experiments with just 6 hours spent per compound. And yeah, it allows us to profile quite large number of compounds in a biological triplicates and to generate very nice and informative data. And these are results we got for the antiviral compounds. The heat map I showed before on the slides, again, the compounds shown in the rows and the proteins in the columns. And on the top, you can see the volcanoplots for several antiviral compounds. And here, the proteins to the right correspond to the proteins stabilized in the cells upon the incubation with the compound. Proteins to the left correspond to the destabilized proteins upon the treatment. What we found out that remdesivir, which was quite on the hype that time, end of the spring, beginning of the summer, was very specifically destabilizing the protein called TRP13. And TRP13 is an essential in spindle assembly checkpoint and played a key role in regulating chromosome recombination. And we found out that this destabilization of TRP13 is very specific for the remdesivir. It doesn't happen in any other antivirals we profiled along with, and it didn't happen to any other compounds which we profile out of these 200 compounds. What is also interesting, in the same study, we included the active metabolite of remdesivir, which is this GS compound. And that compound didn't show any binding events in the cells at all. And this makes sense because the remdesivir in the cells undergo the metabolite to produce the active metabolite, and it could be that this metabolite which we use alone had problems entering the cells because the incubation time we used were quite short, 1 hour incubation. We also ran the antibody Western blot analysis to confirm this binding, and we've seen a nice destabilization of TRP13 in the Western blot experiments, as you can see to the top right. But what is interesting, when we perform the same experiment with remdesivir treated lysates of the same cells, we don't see any stability shift at all. So indeed, the remdesivir needs to be activated in the living cells in order to bind the TRP13. Also, from our observation, the direct binding events of the compounds more often cause the stabilization of the protein rather than destabilization. And in this case, the destabilization of TRP13 might be due to the disruption of this multi-protein complex, because TRP13 forms the hexamers in the cells. And if the remdesivir caused the disruption of this complex, it would explain the destabilization of the proteins in the cell. Yeah, so this is the whole story. And to summarize, yes, Evosep 1 is working fantastic for the TMT-based deep proteomic profiling. It's robust and works pretty much 24/7. And thank you for listening.